Abstract. Our lab is working to develop a non-invasive optical biopsy technique for skin cancer in live tissue utilizing both metabolic and architectural measurements. These entail metabolic fluorescence lifetime imaging microscopy, or FLIM, of NADH and flavins, as well as second harmonic generation, or SHG, measurements of collagen in live mice. Hypothesis. We believe that as the skin of our SKH1 mice is exposed to chronic UV, the metabolic rates of their epidermal keratinocytes will result in increased NADH free-to-bound ratio. Collagen remodeling in the formation of the tumor microenvironment will produce a higher SHG forward-to-backwards ratio. These would indicate an increase in glycolytic energy production and collagen density, respectively. Figure 1. Skin cancer is the most common form of cancer and is usually caused by chronic UV exposures. Early diagnosis is essential for treatment. However, current diagnostic measurements are invasive, leaving scars, and can be unreliable. A less invasive and more accurate diagnostic test would improve prognoses for skin cancer patients. Figure 2. As cells become cancerous, the Warburg effect leads to an increased rate of glycolysis and the TCA cycle compared to the electron transport chain. This results in more free NADH compared to bound. Because free and bound NADH have different fluorescent lifetimes, 400 picoseconds and 1 to 4 nanoseconds respectively, a measurement of fluorescent lifetimes can provide the free-to-bound ratio as a metric for metabolic oxidative states. In addition, cancer results in collagen remodeling during the formation of the tumor microenvironment. We measure collagen architecture using SHG scattering as a second diagnostic indicator. Figures 3 and 4. Here we can see the beam path of our microscope. Notably, we can measure NADH and flavin emission as well as collagen-derived SHG concurrently. In Channel 4, we can also take SHG measurements confocally, which Han et al. Has suggested will allow an estimation of SHG forward-to-backwards ratios in vivo. Figure 5. Once we have fluorescent lifetime images, we analyze them using a Phasor analysis process as shown. Pixels left after ROI selection and thresholding are mapped using sine and cosine transforms to the universal circle based on their fluorescent lifetimes. We analyze this data along two axes. The horizontal axis runs from free NADH on the right to bound NADH on the left. The more vertical axis was identified through analysis of 26 weeks of experimental data from 2019, and we believe it indicates collagen fluorescence. Figure 6. Analysis of these FLIM images show how sham cell phasors resemble in vitro cells. UV-treated cells often appear above or below the NADH free-to-bound axis. In the second and third images, the co-localization of NADH and collagen can be observed, which we believe causes phasors to move along the collagen axis. Figure 7. SHG signal is composed of forward scattering and backward scattering components. In excised tissue biopsies, SHG is measured through two detectors to find the forward-to-backwards ratio, which is strongly indicative of cancer development. In vivo, we can estimate the forward-to-backwards ratio with a single detector by measuring the change in SHG intensity as a confocal pinhole is systematically varied. We can fit the image intensities to the integral of the function S of R. This provides information on the proportion of forward scattered light to backward scattered, that is, the forward to backwards ratio. Figure 8. After our mice were culled, we took skin samples and made histology slides. A skin sample from each mouse was then stained with either a hematoxylin and eosin stain or with DAPI and P53 stain. Changes in epidermal thickness and P53 prevalence were quantified below. Figure 9. Our analysis of the slides included measuring the epidermis thickness. We found that UV-treated mice had a thicker epidermis than sham mice. UV-treated male mice had significantly thicker than female mice. The epidermal thickness did not vary based on sex within the sham mouse group. P53 staining of skin samples was significantly upregulated in UV-treated mice compared to the sham-treated mice. Tumor images from UV-treated mice displayed greater P53 variants and were also significantly different than sham mice. Sham, UV, and tumor images did not have any significant differences based on their sex, unlike the epidermal thickness. Figure 10. Changes in cell morphology can be observed in the raw images and Phasor analysis as well. The tumor images on the left display a higher collagen index than the non-tumor UV cells on the right. Figure 11. Over the course of our 2019 experiment, sham and UV-treated metabolic states could be differentiated from one another. UV mice had a significantly lower NADH bound-to-free ratio than sham mice. This shift coincided with the development of tumors, which indicates it holds promise as a diagnostic tool. Along the collagen axis, no significant difference between UV and sham cells were observed. However, we did note some variation with respect to time. Figure 12. Finally, we can see preliminary results of our SHG forward-backwards measurements. The UV mouse has the lowest forward-backwards ratio, while the sham mouse has a higher value, and the urea crystals are higher yet. However, far more measurements will be needed before any conclusions should be drawn. Conclusion. Our experiment aims to distinguish healthy mouse skin from cancerous and chronically UV-exposed mouse skin through the measurement of the NADH free-to-bound ratio, which indicates glycolytic metabolism, and the SHG forward-to-backwards ratio, which indicates increased collagen density. So far, our experiments have been promising, though we intend to continue our current study into next summer, after which final results will be made available.